detect the differences. Hope I didn't get off target with respect to your question.
Great, thank you.
Laurent?
Thank you, Simone. Thank you for your talk. So you kind of predict upregulation of your genes when you detect splicing events. And what happens, and you showed that in the figure, if I got it correct, but have you cases where you see splicing, but you don't see an upregulation of your genes later on? And could you somehow use this information to infer some regulation, kind of post-transcriptional regulation event?
So you're talking more about like the assumptions that we have, right? You moved away.
Sorry, could you?
So I mean, you're talking more about like if we can somehow validate the assumption that we have that a difference in the relative abundances corresponds to the difference in the actual regulation of genes, right?
Sorry, had a question.
No, I mean, okay, maybe can you re-say it?
I don't know, maybe my question doesn't make sense, but if you see things that don't change, there might be a good, maybe you lack statistical power. There might be many reasons why you don't see what you expect, but there might also be biological reasons behind the scenes.
So one thing, so the thing is that it's a bit like, if you like, this is connected. This is not like RNA velocity, but this is connected to the idea. Okay, I'll stop sharing so we see each other better. This is connected to the idea of RNA velocity, right? So you study the relative abundances of splice and splice reads, and you understand what's going to happen in the short-term future. So the thing is that in order to validate that, you should actually collect time series data, right? Because you actually don't know what's going to happen in the short-term future unless you collect the data. So for a validation, that I think would be possible, but it would actually be quite challenging, right? Because you would need to have, time series data of basically repeated measurements, and then you could see whether when you predict an upregulation, then there really is an upregulation in the spliced mRNA. I mean, that's actually a very good point. I didn't think about it. I don't know how practical that is, but yeah, I mean, you would actually need.
I was under the impression you had that data because I thought I understood that figure.
Yeah, no, we don't have time series data, it's just snapshot.
Thank you.
Yeah, thank you for the question.
Okay, thank you, Simone. I think we are going to pass to the next talk because we are already out of time. Thank you very much.
Thank you for the attention.
So now we are going to hear our second speaker with Beatriz Calvo-Serra. She comes from Barcelona, and she's going to talk about the analysis of transposable elements. So you can go, Beatriz.
Can you hear me?
Yes.
Okay, perfect. Well, I'm Beatriz Calvo. I'm a PhD student at Universitat Pompeu Fabra, and I'm going to present the atena package for the analysis of transposable elements. So transposable elements are DNA sequences that had, or at least had in the past, the ability to move within the genome. In humans, they occupy about half of the genome. They can be divided into two classes. We have class 1 transposable elements, which first transcribe an RNA copy that is then reverse transcribed before inserting in the genome. And then we have class 2 transposable elements, or DNA transposons, which directly excise from the genome and then reinsert in another region. These can act as promoters, transcription factor binding sites, or repressive elements. They can also act as non-coding regulatory RNAs, regulating gene expression or activating cellular signaling pathways. In humans, the expression of these is important for development, and also there are publications reporting an altered T expression in diseases such as cancer, neurodegenerative disorders, or autoimmunity. However, the study of T expression faces one main challenge, and is that they are repeated across the genome. This causes that in short-read sequencing, reads originating from a particular locus usually mapped to the locus of origin, but also to other TEs from the same family located in other regions of the genome. These are known as multi-mapping reads and are usually discarded, causing an underestimation of their real expression. For this reason, we need to use specific software packages which attempt to count these multi-mapping reads. There are a wide range of packages available. However, all of them are available outside R and Bioconductor, although most of them do interact with some packages, for example, for differential expression analysis. So for this reason, we have developed the Atena package. The aim of this package is to facilitate integration of the T expression quantification into bioconductor workflows for the analysis of RNA-seq data. To do so, we provide 4 different quantification methods. The first 3 methods are already existing packages developed outside R in Python and Perl, which we have completely re-implemented in R. And the 4th method is Atena, which is a newly method developed by us. So each of the 4 quantification methods uses a specific algorithm. However, to implement them in R, we developed a unifying statistical and algorithmic framework, which basically is let R be a set of N sequence reads and F a set of PTs. We use sparse matrices to represent alignment overlapping features in a way that if a read overlaps a feature, the value of the set is 1, and if it doesn't, the value is 0. So once we have the matrix with the overlaps, each method processes it differently. For example, RMAP applies some filtering of multi-mapping reads and only counts those passing all filters. Instead, the other 3 methods use all an expectation maximization algorithm to reassign multi-mapping reads. Here we have a visual representation of the EM algorithm. Here we have 3 Ts from the same family, which have unique reads in green and multi-mapping reads in red. As we can see in each iteration, multi-mapping reads are reassigned, in this case mostly to T1, because it's a transposable element that from the start had a higher number of reads mapping to it. So this is what the EM algorithm does. It reassigns multi-mapping reads to their most likely origin, we could say. In this EM algorithm, each method uses different parameters. So after the EM step, we get this reassignment matrix, which again, each method processes differently. So to quantify the expression with Atena, first we can retrieve the annotations using the annotate function, in which we only have to specify the genome version. Then we build a parameter object for one of the methods. Here we have to input the T annotations, which we have previously obtained, the BAM files, which can be ordered by coordinate or read name, which is an advantage with respect to the original methods. And finally, we can call the T expression quantification method with the previously built parameter object, which will output a range similarity experiment with quantification. So getting T annotations is not easy because there's a lack of unified T repositories. There are multiple databases with different nomenclatures, and this makes it very difficult. For this reason, we have developed the annotate T's function, which retrieves repeat master annotations from the UCSC genome browser, which are the annotations that are most used. We actually added these annotations to Annotation Hub so that we can get them through Annotation Hub. And now all genomes available with repeat master annotations at UCSC are now also available. The annotate T's function also can do some parsing of the annotations. In Atena, we have implemented 2 functions which do basic parsing, but the user can specify its own function. So we used a published RNA-seq dataset of Drosophila to benchmark Atena. We selected these 2 samples, which are control and Peewee knockdown. In Drosophila, the Peewee silencing complex silences T's, so the knockdown is expected to increase T expression. So for running Atena for these 2 samples of 20 million alignments, Atena was faster than the original method in all cases. And as for memory usage, it was quite similar except for telescoping antenna that used a higher memory. If we compare the quantification of the original methods and atena, we see that the methods in atena have a high accuracy. And finally, in order to benchmark the new method of atena, we use a simulated data set. We have simulated expressions in 50 loci from 2 different air families with expression levels between 30 and 300 counts. Since the annotations can be incomplete, there can be expression from outside the loci. The atena method and also the telescope method address this by assigning these reads to a category named no feature. For example, in the telescope method, they use a single no feature. This causes that when there's a high percentage of multimapping reads that map outside the notations, in the end step, a lot of reads are reassigned to this no feature. For example, as we can see here, we simulated 0 reads coming outside the notations, but still telescope assigned approximately 1,000 of them to no feature. Instead, in the atena method, we use a multiple of features, and this prevents this massive reassignment of multimapping reads to no feature. As you can see here, estimations are now closer to 0. So as a conclusion, we can say that the atena package reproduces results from the original methods with high accuracy, that the atena method provides a more accurate expression quantification addressing transcripts that are outside the annotations. And as for future work we would like to do is to benchmark the different methods between them. And for the atena method, we would like to implement a method to obtain informative priors from external data such as chipset and use these priors in the end step to obtain a higher precision in key expression levels estimation. Finally, I would like to thank Robert Castelo, my previous supervisor, for his help developing the package and his ideas, and also Pablo Rodriguez, who also helped making atena faster. Thank you.
Okay, thank you very much. Does anyone have a question? Maybe I could start by a question. Could your method could detect, for example, some chimeric transcript in which you would have a promoter coming from a transposable element that becomes activated and that activates a gene that is downstream? Would you detect that kind of
So since we are using annotations from these and genes separately, we wouldn't detect this, but what could be detected is if the T is in the annotations, we can kind of find if there's correlations in the expression of a gene and a T. And if there's a correlation, it's because maybe that's a chimeric transcript, but we cannot detect the chimeric transcript.
Okay. And just another question. This is for RNA sequencing. Could it be applied for whole genome bisulfite sequencing to be able to analyze? I don't know if it exists actually, or
I'm not sure if it could be. I'm not sure if it could be used because you would need the annotations.
I don't know. But not directly, of course, because it's not the same types of reads, but the same method.
Yes, I guess so. With some adaptations of the method, I think so.
Any other question? Okay, so I suggest we pass to the next talk. Thank you very much. We can.
Thank you.
Hello.
Hello. So
You can hear me now.
So now we are going to hear Alexandru Mahmoud, who is going to talk about yes for cure. So
Thank you.
no, oh sorry.
All right, it's
Okay.
Vince Carey presenting. Alex is not presenting.
Okay,
this,
so it's
but I
Vince
will.
who is going to talk about
Okay,
Yes, for okay.
all right.
You
So let's see if I can find the right tab. Yeah, here we go. Well, thank you very much. I'm understanding this is a 10-minute slot and that we're a little behind schedule. I'll try to be brief. Please interrupt me at any time if there's a question or a need to stop. This is live, github.io. It's a package down site called Yes CDS. Yes is Young Empowered Scientists. CDS is Cancer Data Science. Young Empowered Scientists for Continuing Undergraduate Research Experience is a program at the Dana-Farber, and we produced this with the help of the Chan Zuckerberg Initiative Diversity and Inclusiveness program to get some folks from a high school undergraduate population and attract them into some data science questions about counting cancers, building maps, studying the anatomy of cancer, and understanding clinical trials. And this was part of the advertisement that was used to attract students. There's about 100 in the program. We estimated that 5 would come and join, 11 did. And they came for 5 meetings over the summer to talk about these various topics. And the issue I'm trying to address in this talk is what Alex Mahmoud has built. Out of this package, the authoring of these materials, injecting them into a cloud computing framework called Jetstream 2 under a Galaxy framework. And just to put the subject matter in perspective, I have this little chapter of our package down site, which I just made this morning. This is from a paper called, I don't know, making a more inclusive genomic data science community. And there's a project called GDSCN, the Genomic Data Science Community Network. And the idea is in the United States, there are institutions, maybe I need to blow this up a little bit so it can be seen better, institutions that they call underserved. And they include community colleges or historically Black universities, Hispanic and tribal colleges. And in order to grow the workforce, it's really important that a couple of things be accomplished. The first, that there be outreach to individuals at various points in the educational sequence. And the other is that it be equitable, so that individuals from many different ethnic backgrounds, socioeconomic situations, and so forth, have the opportunity to get engaged with genomic data, which, as we know, is exploding. So there are the R1 institutions, Research First, and the UI, and the idea of the GDSCN is to get these folks together and have them learn how to teach a much broader collection of students who can do genomic data science. And it was pretty obvious to us that Bioconductor would be a useful tool in this. This little schema says, you know, we're usually taking folks who are doing genome biology or those who are good at high dimensional data science and trying to bring them together. They all have to face up to the things that biotechnology is producing in terms of, you know, massive volumes of heterogeneous data. And Bioconductor has been around for 20 years, helping people to get that stuff into their computers and do some analyses that ultimately get us in the space of where we want to be, which is inference on biological questions. So that is true. Bioconductor can help with that. And this little slide is just telling us, well, this was the old version, that R packages and R Markdown can be regarded as instructional assets in this process of building inclusive genomic data science educational materials. And so what I'll be showing you live in just a moment is this cloud-based, but it's a public cloud. So this is not something, this is something that comes from a grant of resources from the National Science Foundation, where we have deployed Galaxy, but also specialized it to providing material for this Yes for Cure project. So why don't I jump over to that? Mm-hmm. Come on now. I'm having trouble changing my tabs here. Okay. Well, let's do it from scratch. How am I doing? Here it is. So this is a Galaxy instance, and these are chapters for the components of the workshop. So you click on one of these, and we trained the students in advance that in order to work with this, you're going to have to click on this, and you're going to get a Galaxy interactive tool, and we'll ultimately be using Jupyter notebooks. We felt that that was probably an easier activation energy framework for this than RStudio would be, which has all its many panels and so forth. So you wind up having to start up Jupyter Lab, and this is a personal Jupyter Lab running in this cloud. And in order to do chapter A0, you will click on that. And now you have a Jupyter notebook, which takes us through. This was an introductory notebook where we try to get a sense at the outset of what the individual's understanding of various aspects of cancer data science is. And so, for example, you see some survival statistics collected by the SEER program. You see a histogram. Can you interpret this histogram? What's going on here in terms of rates collected over time? And these are questions where we just ask, you know, can you interpret these now? And then at the end of the program, we hope you'll be able to interpret them better. I'm going to close this down and start one of the other ones. So we can just go back to Yes for Cure now. And just look at one of the other chapters before I leave finish the talk. And that's C3, molecular origins of tumors and their spread. So the idea is that we start with some very basic issues about how you collect rates, what institutions collect rates in the United States, and then we actually plot these against geography. And finally, we want to get into some of the molecular details and then think about treatment in clinical trials contexts. And so this C3 is about molecular origins. And once again, we have to jump through a couple of hoops to get to where we want to go. We're going to click on C3 over here, and we have our notebook. And so we can take the students through the basic ideas of the human genome, the basic dogma, central dogma, and then start thinking about data science problems where you have thousands of features of potential interest over these chromosomes, and then you start to drill down and understand what could be affected by a mutation in a given cancer. And so we take them ultimately through a number of things that are easy to get using cloud level genomic resources. And we wrap up with this idea here, which is an individual treatment program for cancer in the case of a metastatic breast cancer. So I'm going to stop here because it is this level of data that was really of immediate interest to the students. And we didn't get there for a while. And I don't think many of us get here. We do things with TCGA and so forth, but you know, nowadays you really need to be able to go deeply in multiple modes to do this. And so the question is, how can we best facilitate teaching in this space? Can we make it easier? Can we make the data be real? Can we help the authors to get the material into a framework like this? I think we can. It's all done using R Markdown and GitHub and other things that we're all familiar with. And so I'm happy to talk to you about that if you're interested individually. The package, Yes CDS, has its own GitHub site and issue filing and so forth. So I thank you very much, and I'm happy to take any questions. I'll stop sharing so we can see one another.
So thank you very much, Alexandru, Vince. So if anyone has a question, will come. Please come.
Hi, thank for the very nice talk. I was just wondering, for this entire genomics data science outreach program, is it limited to just cancer, or does it also include other conditions?
The GDSCN is going for the whole panoply of problems in genomic data science. We picked this because we had an undergraduate population that was already focused on cancer and doing wet lab work, some of whom might be interested in doing data science, none of whom were required to do it, and many of whom will not be doing it in this program. So we want to see whether this is feasible in this context.
Okay, cool. Very simple answer. Thanks very much.
Yes, Davide. He's coming. He's coming.
Hi, Vince. Very nice system. I was wondering to what I mean, this is a really great program, but I was wondering to what extent this is sort of like extendable or generalizable to the many courses that we do at the various levels.
Yeah, thank you, Davide, because our intention is to be extremely flexible and inclusive and redeployable. So that's where Alex has really made something through Helm charts that you can pick up and plop on, a suitably equipped Linux system with Kubernetes, and you're ready to go. And so what we're doing here is we're putting vignettes in an R package, and we have a makefile that just produces the Jupyter notebooks out of them and puts them in the place where they need to be so that that Galaxy instance can present them to the users. So, there's a little bit of specialization there. We're trying to weaken the requirements for each specific technology. So, for example, Quarto is a way of having multilingual teaching materials. And I think the other thing to break away from is the R package, because that's a little too complicated. What if you just have something sitting in GitHub that's an R Markdown? Why shouldn't you be able to teach from that? And Alex is working on facilitating all of those things.
Fantastic.
Okay, thank you very much. I think we're going to continue because we are quite late.
I know. Okay, thank you.
So now we are happy to introduce Helen Lindsay. I don't know if you hear us. Yes, she's going to give us some tools for the site-sec pre-processing.
Okay, so sorry I couldn't make my workshop yesterday, and thanks to the organizers for giving me this opportunity to present. SiteSeq is a technology that allows you to simultaneously measure. Is everything all right? You're all hearing me?
Yeah, we can hear you, but
okay,
we don't
great.
see the screen.
So SiteSeq is a technology that allows you to simultaneously measure antibody, cell surface protein expression and gene expression. So the way it works is that an antibody gets conjugated to a sequencing oligo. The antibody binds to its cell surface. Yes?
You need to start sharing your screen. We can't see your screen right now.
Did you see it at all?
No.
No, not at all.
Not at all. I'm really sorry. From my end, it looks like I'm sharing.
Okay. Yeah. Okay. 1 second. Let's kick him out. Sorry. That one.
All right. Do you want to go to Pierre?
So can you start sharing your screen again, please? Oh God. Probably she's reconnecting, I'm not sure. Oh yeah. Oh. I think we have a minute. Oh yeah, now we can see it. Thank you very much. Great.
Okay, so start again. No?
Yeah, if you start from the beginning, that would be fantastic. Thank you so much. So good. You need to unmute yourself and start your video again, please. And then we are all set.
You only have 3 minutes left.
Yeah, so that's unmuted now and video started. So we're getting there. Great.
I'm so sorry.
No, was not your fault. Thank you so much.
Have you got everything now?
Yes.
You can hear me?
Yes, we can
see
hear.
me?
Everything works all right. You can start. Thank you.
Okay, so I'll start with some acknowledgements. This is a project led by Rafael Gattado, and it's a collaboration between his team at the Fred Hutchinson in Seattle and us at the SHU in Lausanne, with funding from Chan Zuckerberg, NIH, and University of Lausanne. Okay, so this is part of our efforts to curate a large set of publicly available CiteSeq data and make it easy to compare across studies. Bernard has been doing the data curation, and if you didn't get a chance to see his poster in the last session, then I would encourage you to check it out. Ye Zhang at the Fred Hutchinson Center has an R package ADT norm for normalizing the protein component of CiteSeq data. It's inspired by methods from flow cytometry, and it works by aligning negative and positive peaks. It's particularly useful if you want to normalize across samples and you don't necessarily have empty cells or isotype controls that you might use for detecting background antibody binding. So her method is available as an R package on GitHub. The preprint is available on bioRxiv. So for the rest of the talk, I'm going to talk about my package AB names, antibody names, which is a package to enable you to standardize protein names in a CiteSeq experiment. It's available on GitHub, and I'm aiming for the next bioconductor release. So the problem with antibody-derived tags is that there's no consensus for how we should report these. So you could report, for instance, the antibody, but you could also report what it binds to. And this is an example of the same protein reported across different studies. On the left, you see the antibody information. This is reasonably standard because it comes from the vendor, and there's only a couple of vendors. But then in the center column, you see what was actually reported in the data. So you see everyone has a different idea of how these things should be reported, and that makes it really difficult to be able to say what's the same protein across different studies. So what I've been doing is to try and map antigen names to some sort of stable ID. And there's a data set in the package, gene aliases, to help you do this. What sort of stable ID should we use? Well, it really depends, because antibodies can bind to all sorts of things. They can bind to a protein, a multi-subunit protein, an epitope shared by several proteins in a family. They can bind to a specific allele or an isoform, and sometimes they bind things that aren't proteins at all, like sugars or lipids. So what I've done is, starting from the Human Genome Naming Consortium, I have collected gene aliases from a number of different sources, and also protein aliases from different sources. And in the instance that this was not enough to enable me to map from an antibody name to a stable ID, then I did some manual curation using the package roles to look up in ontology services. So a few important details. HDNC is the reference for all of the IDs. So I start with HDNC and then I map things into that, and ambiguous aliases are removed. So it's very often the case that the same name refers to different proteins. So I've taken these out of the data set. As an example for the isoforms of CD45, this is part of the gene aliases table. You see that they all map to the same gene, but the gene is not enough to enable you to distinguish between the different antibodies. So in the gene aliases package, in the gene aliases data set is an alt ID, and this can be anything. In this case, it's an ID for the isoform, but it's anything that enables you to distinguish between different isoforms. And the final column, source, tells you where did this information come from. So in this case, it was manual lookup, meaning that I looked this up myself in roles. But we still need to be able to go from the name as it was reported to something that you can match into the aliases table. So to do that, I have the function make query table. What this tries to do is, starting from a name like the one on the left, pick out the parts that might be the gene name, and using various regular expressions, try to format them so that it matches a gene name. Now on the right column there, you see the query terms that this has generated. Some of them are more sensible than others. But if we use the function search aliases to then search in the query table, we come up with the single best result. So what search aliases does is to match antibody names to the gene aliases and check that all of the matches are coming from a single entry in this table. Unless, of course, we expect multiple matches, for instance, in the case of TCR receptors. So that is enough to enable us to say which antibodies are the same. Still, the question still remains of what we should name them. So I don't actually have an opinion of what these should be called, other than that they should be consistent. So I've gone with democracy, and I have collected antibody information across a number of studies, and I've looked at what is the most common name that's given first. So for this example, this would then be called CD274. So AB names is a package that has data sets to enable you to map between antibody names and a standard identifier. A lot of information from other studies about what the antibodies were called, their clone numbers and catalogs numbers, information from the vendor BioLegend, and some specialized matching functions for matching and viewing and checking the results, and enabling you to take a site-seek study and match the names to our consensus. So thanks for your attention. Sorry for the technical difficulties. Raphael is recruiting. There's 2 positions available. So if you're interested, keep an eye out or send him an email. And thank you.
Okay, so thank you very much. Any questions? No questions. Okay, so as we are a bit late, we might pass directly to the next talk. Thank you very much. So we can start again. And we are going to listen to our last speaker, who is going to talk about epigenomic data analysis. So we are going to listen to Pierre-Luc Germain.
Can you hear me well?
Yes.
Okay, thank you. And you see the screen?
Yes.
Okay. So good afternoon, everyone. I'm sorry I couldn't come due to COVID, but thanks for giving me this opportunity anyway. This won't be about single cell, but about rather classical stuff. The project started basically last semester when I was teaching basic epigenomics data analysis, so stuff like ChIP-seq and ATAC-seq analysis. And I couldn't help but feel that my workflows were kind of unsystematic, and I wanted to try to build some resources that would be a bit, I don't know, a bit more systematic and a bit smoother for students to use. And although there is a related package, this is at the moment very far from finished. And the aim for today was rather to get feedback from the community before going further. So I'll keep this very brief and end up with some questions on which I'd very much appreciate some feedback. Okay. So a rough general epigenomics pipeline is going to typically look like this. People are going to do some quality control and trimmings, align the reads. Then typically we generate bigwigs for the purpose of visualization. Then some peak calling. Max is still very often the standard. And then for the downstream stuff, we typically rely on R-based tools, or for visualizations also deep tools. Now, when we try to look for R alternatives for this, for some of the main steps, so especially the first two steps, I think there are very good solutions that work very nicely. And for the downstream stuff, there's a wealth of really useful packages to handle that. But there are some gaps, especially things like bigwig generation and peak calling. As far as I know, there's no really convincing package for that in R. And so, of course, I thought it can't be that hard, and I went down the rabbit hole of trying to fill those gaps. But it's not mainly about filling gaps. It was more about actually ensuring some kind of interoperability and easy back and forth between those different steps and between stuff that we do with different programs and packages. And I'll just give one example of a problem that I've often seen in the work of students. So when we work with bulk data, we typically have BAM and bigwig files, and we use these different files for different purposes. So for example, we're going to extract peak counts to do some differential binding or differential accessibility analysis. We're going to extract profiles in order to plot patterns and maybe cluster regions. And we're going to also use the data to quantify, or to estimate some kind of motive activity or footprint activity and so on. And if we look into each of these steps, actually, most of them include some kind of normalization. And very often, actually, there are different kinds of normalization, which leads to inconsistencies. Because, for instance, with the peak counts, people are typically going to use a standard peak TMM-based normalization, for instance, whereas for things like the plotting profiles and clustering, background normalizations or coverage normalization are more common. And of course, if we then use different types of normalizations with different types of downstream analysis, we're going to end up having some inconsistent results. Well, often they will agree because the normalizations will do the same things in many scenarios, but often they don't. And therefore, it would make more sense to have general implementations of the normalization methods that can work on any kind of these inputs, and the output of which can basically be input into any kind of downstream activities, downstream analysis. And so this is just an example of the kind of smoothness that I felt was missing, at least from my way of working with epigenomics data, especially in comparison to other fields like transcriptomics. So the aims were to make this type of analysis smoother. And the sub-aims were basically to try to fill some gaps, R gaps, so to speak, in the workflow. The second was to enable this sort of easy interoperability, and that equivalent operations can be performed in different contexts, such as plotting and differential analysis. And for me, two main types of operations were especially normalization and bias correction. So GC bias correction, but also in ATAC-seq fragment length, different distributions of fragment lengths across samples that need to be corrected for some purposes. But hopefully this could be then generalized to something else. And then the third point was to facilitate visualization. I often work with biologists and giving them the tools in order to be able to look at the data themselves and to plot a lot of stuff by themselves is actually, as far as I'm concerned, a very productive way to interact with biologists and for students to get a handle on their own data. Okay, so I'll just give a quick idea of some of the work that I've been trying to do in this direction. So first about filling some gaps. So actually implementing a R-based bigwig generation function is not very complicated. But what's very nice, I think, is that if we try to do all the operations, including the tiling at the level of the run length encoding, for which the bioconductor team has developed super efficient methods, we can actually make this pretty efficient. And this is just a comparison of the same task done in deep tools and in R, and you see that it's actually considerably faster in R than in deep tools. And this is specifically because of the tiling that's done at the run length encoding. Right, obviously the function has many different parameters that allow us to generate different kinds of signals that then provide different kinds of information. And then the downstream aim, of course, is to visualize them and eventually use it for clustering and the likes. This is an example of different such signals. And of course, the efforts were also to try to offer a very simple interface around enriched heat map, especially to handle the normalization and the trimming that needs to be done across different experiments. The interface, yeah, as you can see in this example, code is very simple, but the workhorse underneath, of course, is just the enriched heat map package, except for the reading of the data and the tiling again. Okay. There are also wrappers for GViz, but I won't get into that. It's simple enough. Peak calling, this is very preliminary. Again, here the aim was to try to do roughly the same thing Max is doing, but working on the run length encoding. At the moment, all I can say is that there is a high concordance with the output of Max. This is a concordance at the top plot. And that because we're not bin-based in this analysis, it yields much narrower peaks that are better centered on the motif. So nearly an order of magnitude smaller, and yet retaining almost all of the motifs that are found by Max peaks. But again, this is very preliminary. It's just to present a bit the gist of the effort. And I think as when people in the audience see this, probably what you're thinking is that this is reinventing the wheel, and that's of course what I'm thinking as well. So what of course, the attraction to do so is always great. And what I wanted to do through this small talk was especially to get feedback as to this basic question. So does it make sense to work in this direction? Do we even need BioC-based Max or deep tool alternatives? So at the moment, for instance, there's a Max R package that use Basilisk to set up a Conda environment and so on to run Max. That seems a bit heavy-handed, but it does the job. So is it necessary to come up with anything else? Am I the only one that feels the need for a kind of smoother integration between different tasks and different tools for this kind of analysis? Do I miss all the good solutions already out there? And if it makes sense to work further in this direction, basically whether this should continue to be a kind of individual and necessarily idiosyncratic effort, or whether it can benefit more from the community, so has more input from the community in order to better connect with general BioC resources on the one hand, but also to more specialized packages for very specific applications. And I know that not being present in person isn't ideal for getting feedback and discussing this. So if anybody wants to further discuss this, I'd be very, very happy to have chats on the BioC Slack or any other venue. And that's already it. And if someone has some feedback on these questions, I'd be very happy to hear them. Thank you.
Thank you very much, Pierre-Luc. So as anyone has a question or wants to answer the question?
Well, I'm going to jump in if you can hear me.
Yeah.
Very nice talk. I think you may be too modest when you say reinventing the wheel, because you gave some results that seem to suggest that you actually have improved performance. Isn't that correct?
Yeah, I mean, You mean the speed improvement and the likes, or the
you talk about narrower peaks also. So,
sure.
You know, I don't know whether you are able to present these as, you know, definitive methodological advances, but if you are, then I wouldn't be so modest. And I think that it does make sense. But, you know, I'm just one person and I don't actually work in that field, but I think there's a tendency to be extremely modest when you have these wonderful tools that are on everybody's desktop and you say, well, maybe I can do it a little bit better. And everybody says no. But if you did, you should take credit for that.
Okay, thank you, Vince. That point taken.
I mean, do you feel the spectrum of problems that you've looked at is broad enough that you can say, yes, I think it actually is better?
Well, no, for the peak calling, I don't know yet, honestly. I mean, it's just this one point for the moment, but I'm testing, especially I'm interested in peak calling in the absence of an input for broad systematic reanalysis of data from the literature. And there, for instance, I think it's still not as good as max. So there are certainly aspects on which are definitely not better or even final there. But I mean, this is really work in progress. But I must say my aim was not at the beginning to improve upon those methods. But if I do if I do, then I'll definitely take it.
Yeah, so I think the smoothing part is also, you know, in terms of our educational objectives, to
exactly
reduce the number of technologies people have to jump across, as long as you give credit where credit is due. You're using Max2, but you're doing it in a simpler way through R. That's also an advance.
Okay.
Great. So Pierre, thanks for the nice talk. Just a piece of feedback working in the eClip field. I absolutely appreciate our implementation of some of the tools, because at the moment we're wrapping a lot of things in SnakeMake and we have to work with from R SnakeMake and do workflows. And it would make some things definitely easier, at least kind of workflow wise, and definitely make things a bit more convenient for us.
Okay, thanks. That's good to hear.
Okay, Bill. Any other question? No? Or we stop now? Maybe we're already very late. So you can stop here. Thank you very much.
Thank you.
Luc. And okay, so have a nice evening. Don't. Oh, no. No, we'll have a nice evening, but not yet.